Okay, great. We're so pleased today to share some time with one of our favorite institutions, University of Florida in Gainesville, where the University of Florida Diabetes Institute is coming to us live, and they're going to discuss in 3 sessions. First, the clinical trials roadmap. Secondly, we have a talk regarding innovations in cellular engineering. And finally, exploring the whole pancreas and type 1 diabetes. And our first group today, we're so pleased to introduce Dr. Todd Brusko. He is now the new director of UFDI. And we also have Dr. Michael Haller, Dr. Laura Jacobsen, and Dr. Melanie Shapiro. And I'm going to say a little bit about each of them, and then I'm going to let Todd Brusko take it away and kind of walk you through what is happening at UFDI and sort of guide the conversation to new advances, as well as, the kind of, excellent funding and resources that are available there. Okay, so starting off at the top, Dr. Todd Brusko, he's appointed as a professor in the Department of Pathology at the University of Florida. He currently serves as the Assistant Dean of Basic Research for the College of Medicine, and he's the newly appointed director of the UF Diabetes Institute. His research interests are centrally themed around understanding the mechanisms by which the immune system maintains a state of control, aka immunological tolerance, and this includes studies of the innate and adaptive immune system with a particular focus on adaptive immunity receptor diversity and control through co-stimulation. He's previously published over 150 studies reporting cellular immune defects in patients with autoimmune diseases and investigated cellular immune mechanisms and the biological basis of type 1 diabetes. Michael Haller, MD, is currently the professor and chief of pediatric endocrinology at UF, where he coordinates an active research team focused on prediction, prevention, and reversal of type 1 diabetes. He is an active investigator in TrialNet, TEDdy study, and the T1D exchange. He serves as the PI at the University of Florida TrialNet Clinical Center and the chair of the Clinical Implementation Committee for TEDdy. Dr. Laura Jacobsen, MD, is an assistant professor of pediatrics specializing in the care of children and adolescents with diabetes and endocrine disorders at UF. And she's also a physician scientist at UFDI, working in prevention and intervention clinical trials for individuals and at risk for type 1 diabetes and translational projects studying biomarkers of response to immune therapies. She's proud to serve as an investigator in TrialNet, and she's the chair of Pathway to Prevention Screening Program in TEDdy. Welcome, all 4 of you. Thank you very much. Your incredible bios really illustrate the power behind this team. And we really appreciate you taking the time to sort of talk about what's happening at University of Florida in Gainesville in terms of clinical trials and kind of really shining a light on the excellent science and opportunity that exists there.
Yeah, thank you, Monica, and welcome, everyone. I do think we're going to share some slides just so I can give a brief update on where we are in terms of some of our efforts. All right, so thank you. I'm Todd Brusko. As mentioned, I'm a professor here in the Department of Pathology and acting director of the Diabetes Institute. Next slide. So I think one of the unique aspects of UFDI is that it's really a comprehensive group, and we try and cover basic science research, and you'll hear a lot about that later on today with some of the other sessions. Today, we have, I'm fortunate to work hand in hand with some really amazing physician scientists that are actively bringing new therapies into the clinic. And I think what's really unique and what makes us operate as a group is the ability to have really high quality basic science research integrated with medical interventions and try and understand mechanism of action. And then a big component of what we do here in the UFDI is try and train the next generation of both physicians and scientists. So we have educational programs all the way from undergraduates up through fellows that we bring through the labs. So I think you're going to get a little hint of where we are, but this is the Biomedical Sciences Research Building. And one of the great aspects of it is that the UFDI resides primarily up at the very top on the 5th floor here, but we are integrated in the same building with biomedical engineering. So later on today, you'll hear from our colleagues, Dr. Sherry Stabler and Ed Phelps, who are on the third floor. So we directly interact with them on a regular basis. And then we even have our own animal facilities here so that we can do very preclinical early stage immunology studies in the NOD mouse as well. T1D research program here at UF is fairly extensive in terms of the programs that we're involved in. A lot of this early work was sort of led by Des Schatz here at the University of Florida, along with other universities here in Florida, to create intervention trials like TrialNet. We've been involved in key studies like TEDdy to understand the environmental impacts. And then I think what Mark Atkinson founded here at UF is this network for pancreatic organ donors with diabetes, which I think is really transforming our understanding of the pathogenesis of disease and our ability to interact with the entire research community and share those tissues across the entire world. So we have two major biobanks. One is the living biobank, where we try and acquire peripheral blood samples from patients with type 1 diabetes or those are at risk. Those samples come from not only here in Gainesville, but Orlando, Baylor, collaborators at Emory, and then all the way up into Ohio as well. We acquire about 400 to 500 peripheral blood samples a year. And all of those samples undergo sort of rigorous basic phenotyping. So we get genotypes on all of those subjects. We're one of the core autoantibody testing sites in efforts led by Clive Wasserfall here at UF. And all of that gets integrated into our database so that we can draw on those samples and pull very specific genotypes and understand molecular pathways towards the pathogenesis of type 1. So this just gives you an idea of some of the types of samples that we're acquiring here at UF. So we have really the full spectrum of a cross-sectional sample of autoantibody negative controls all the way through single to multiple autoantibody positive subjects. And it really gives us the ability to draw from not only different age ranges, but also different genotypes so that we can pull susceptible versus risk genotypes and really understand molecular mechanisms in this living biobank that we have. So these samples not only come to UF and help support our investigational studies locally, but then we try and distribute some of those samples as well. So we operate a biobank that takes requests for both serum and PBMC samples, and we're able to share those across the country and really across the world as well. I think what's really unique about UF here, and you'll see this in some of the lab walking tour as well, is that we, through the efforts really of Mark Atkinson and Irina and Martha Campbell Thompson and Clayton Matthews, we've been really a site to try and study the disease as a whole body problem. So we've acquired not only the pancreas of organ donors with type 1 diabetes, but other tissues as well. So central tissues, right? Understanding how T cells develop out of the thymus, understanding B cell growth and development from the bone marrow, and then all of the secondary lymphatics. So it's really a comprehensive effort to profile the immune system in subjects with type 1 diabetes. And what makes this possible is this infrastructure that really brings in samples and tissues from all of the OPOs across the entire US. So this is just a quick slide demonstrating some of the key investigators involved in clinical and translational efforts here at the University of Florida. It really does take a team, I think, to do this science at a high level and do these interventional trials. And you've heard about some of those here at the table, but it's a much larger program here that is very well integrated. Next slide. So we're going to take a minute and just show you a little quick tour of the University of Florida, made by one of our staff scientists that has been here with us for a number of years.
As the sun rises in Gainesville, Florida, the animals begin to stir. And the most famous of these creatures is the alligator. This symbol of our city watches over all who pass by, and they are a constant presence at every corner, be it manufactured or natural. And it's seemingly through their eyes that we see our glorious stadium. And the many historic buildings and notable landmarks found here at the University of Florida. Our main campus is buzzing with academic activity on a world-class scale. But as we traverse our natural areas and ford the streams just downhill from our most well-known landmarks, we arrive at the equally high-caliber home of our College of Medicine. The UF Health and affiliated College of Medicine complexes offer an eclectic array of classical and modern facilities. These campuses are enveloped by both bustling cosmopolitan and pristine natural settings, all echoing the long history of studying and practicing medicine at this institution. This history is respected and humbly shared by the Type 1 Diabetes Research Labs at the University of Florida Diabetes Institute. Since before the discovery of insulin, Type 1 diabetes research has been fixated on finding a cure by any means possible. And although the research community has tempered our expectations for a quick fix over the past decades, here at the UFDI we maintain this original fervor for finding a cure. And in our research labs, every door leads to discovery. The UFDI research space is housed in a continuous open lab environment. Here, we perform all facets of scientific investigation into the causes and effects of type 1 diabetes and into possible therapies for the disorder. Integral to all of these research activities is scientific equipment, which is contained within our labs and in our core facilities. We utilize cutting-edge instrumentation as well as standard scientific equipment found in many immunology and cell biology labs. These devices are ever present as we traverse our labs because they are, of course, critical to our science. But more critical still are the samples we receive through initiatives like NPOD. In our biobanks, human blood and organ donor tissue are received and thoroughly processed, then separated into smaller, sometimes microscopic components, which are stored locally until they are ready to be prepped for shipment and sent out to other diabetes research labs all over the world. All the activities involved in processing samples and generating data can be dizzying. So after the experiments are complete and the results are ready, where do we turn? To our analytical experts. As technologies have advanced, it's been increasingly necessary to supplement our experimental laboratory skills and data collection with equally capable data management and analysis expertise. Utilizing current software and leveraging UF resources like the powerful supercomputer called Hypergator, we are able to visualize, analyze, quantify, and report statistics on multitudes of experimental results. And these results, in turn, send us back to the lab, where we put our minds together, address any lingering questions, and come up with more experiments that hopefully help the entire diabetes research community get closer to the definitive T1D cure that has been elusive for centuries. This is the UFDI Research Lab, and amongst all our facilities and cutting-edge technology, it's surprisingly easy to find the key to our success. As our work pauses each day and the sun begins to set over the University of Florida, we must once again leave ourselves to the watchful eyes of the gators that never sleep.
Wow, that was so impressive.
Yeah.
I mean, if that really makes one, want to just run, don't walk to UFDI if you're interested in type 1 diabetes research. I mean, just the vibe, the beautiful campus, the seamless, connection of the wet lab to the quantitative analytics, the open lab space, the whole team science approach. This is really very, it's really compelling to think about UFDI if you're a young trainee or early in your scientific career. I would say it's just definitely one of the top. So I would like you all now to sort of everyone's sort of thinking about that beautiful video. I don't know, Todd, if you want to talk a little bit about strategies for moving forward to clinical translation from basic science discoveries that are emerging from your basic science work in the biobanks.
Yeah, so let me quickly sort of touch on, I think, a key strategic area for us. One is that biobank that we've been building for decades, from the efforts of Des Schatz and Mark Atkinson. I think one of our strategies is to, when we understand genetics, I think our chances of getting on target hits are going to be much better. So I think we spend a lot of time trying to understand genetic risk variants and how they impact immune regulation, and thinking about drug development and discovery on those targeted pathways. So that's one big area of focus for us currently. The other area is that, we have this amazing tissue biobank. And I think for the first time, over the next 5 years, now that we're able to do really in-depth spatial biology, I think we're going to have real signal for what is going on within the pancreas and the pancreatic draining lymph nodes and secondary lymphatics, and really be able to find key targets that we can address and really direct therapeutics towards. And then obviously, Mike and Laura here are actively involved in intervention trials with agents like ATG and many others that they are involved in from other industry type collaborations. So that's going to be a big area of interest as well. But I'll turn it over to you guys. Yeah.
Thanks. Laura, I wanted to ask you, can you weigh in a little bit regarding your thoughts on the different treatments based on stages from your work in TrialNet, the TN01 natural history?
Yeah, so that's definitely an evolving area of type 1 diabetes. So, right now we have the staging system that was developed in 2015, looking at people who are at risk for or who have early stage type 1 diabetes. So just multiple autoantibodies with normal glycemia or multiple autoantibodies with dysglycemia. Those are stage 1 and stage 2. And so we've learned a lot since 2015 when those stages were developed. And I think it's great that TrialNet has been able to do so many trials in stage 1 and stage 2 because these are really difficult populations to recruit for. It's a difficult concept to explain to families and even to other healthcare providers. But definitely it's been fruitful because it's resulted in the FDA approval of teplizumab. So we were part of that clinical trial, and that now gives an option for families who are very high risk of developing symptomatic type 1 diabetes or stage 3, that they have a therapy that could potentially delay that onset. So we're continuing to look for people in TrialNet in the pathway to prevention who are at risk, who have antibodies that are a marker of type 1 diabetes. And so what we really want to do, though, I think expanding on the stages. So the stages are a nice framework for us to communicate about early stage type 1 diabetes with the lay public and with other scientists. But the stages really identify a really broad, specific risk of progression to stage 3 type 1 diabetes. So we do a lot of work here trying to understand different metabolic, immune, and genetic markers that can identify those risk features. And I have a slide that I can show that kind of illustrates that, and then probably can let Melanie talk more about it. But what we have are basically these original stages that are very kind of simplistic. But if you look at this figure up here at the top, these represent very wide or variable amount of actual disease progression. So if you were someone in stage 1, if you just use a stage, you could have either a 6 or an 84% chance of progressing to stage 3 within 5 years. So our group here, in collaboration with other people outside the University of Florida, so myself, Mark Atkinson, Steve Gittleman, and Jay Sasenko, have published a paper trying to actually push the field into more of a personalized disease approach, which has been a real forefront that TrialNet has been pushing as well. So instead of saying we have these kind of non-specific stages, we want to use all the data that we've collected on individuals who are in these early stages of type 1 diabetes. And that's Melanie's genetic data and Todd's immunophenotyping data and Mike's metabolic data to try and understand a single person what their likelihood of progression in a set time period is, so they know whether they want to enter a clinical trial or should they get an FDA approved therapy. So that's a lot of what we're doing with the stages of type 1 diabetes. So it kind of goes towards personalized risk.
Yeah.
And that really feeds in nicely.
It's also reminiscence of the evolution of the breast cancer field where, at one point it was one and done, and then they became more personalized and they're targeted with their therapies. So it's not like this doesn't have a roadmap in other disease states, right? You guys are really following that ideology and it's been very successful in breast cancer, for instance.
Absolutely. And talking about disease specific interventions, so like precision immune therapy treatment for type 1 diabetes. So, we do a lot here with anti-thymocyte globulin, as Todd said. And so we've actually looked at several clinical trials that have been done with ATG in people with newly diagnosed type 1 diabetes. And we can identify who are the most likely responders so that we can target these immunotherapeutics to the right people in the future. So some of the work we do is trying to identify those responders ahead of time before a drug is given. And then we can actually do what's true precision medicine, where we say this person's coming to us now with whichever stage of type 1 diabetes, we recommend they get this therapy, ATG, teplizumab, rituximab, whatever it may be, based on their immune profile and how that drug works. So I'd love to hand it over to Melanie to talk about her work that's, I think, really novel, looking at the genetics of predicting, because genetics are immutable, right? Like they actually could help us predict who should get what therapy. Go to your slides.
Thanks so much for that. Yeah, Melanie, can you weigh in on your work and contributions thus far?
Thank you, Laura. So a lot of my work has been focused more recently on using genetics to predict who will respond best to immunotherapies for type 1 diabetes. And a lot of this work is retrospective analysis, analyzing previous clinical trials from both TrialNet as well as Immune Tolerance Network, since there has already been a ton of both genetic and immunological data that can really be delved into further. So here we're looking at a figure actually from a review that we just published with collaborators at Children's Mercy, Kansas City, where our collaborators are experts in artificial intelligence and machine learning. And we partnered with them to propose ways that we can use AI and ML to better predict who might respond to these type 1 diabetes immunotherapies. So we came in as sort of the immunology experts and they came in as the AI experts. And together we formed this proposal where one of the things that we can use to predict responders is genetic variants. And so here we're looking at different genes that are known to contain many different mutations that are associated with differential response to therapies in other disease contexts. And so many of these therapies have been repurposed for type 1 diabetes, but even though we know that these gene variants affect response in, for example, things like cancer, other autoimmune diseases, and transplantation, these variants have not been studied in the context of type 1 diabetes for many different reasons. One of the big reasons being that our clinical trials are quite small, and so it can be hard to find signal. But one of the really nice things is that a lot of these variants are very, very common. And so even when we look in these very small trials, we have a chance to see some sort of signal. So here we're looking at a couple of different pathways where there are variants associated with differential responsiveness. So one being, of course, variants that affect metabolism of small molecules. So you can see some of these enzymes involved in drug metabolism are highlighted on the picture of the person. But then when we zoom into some of these other areas, there are some other targets that we might be able to look at. For example, variants that affect the mechanism of action of antibody-based therapies. So for example, there are lots of variants, both copy number variants and single nucleotide polymorphisms associated with differential response, for example, to rituximab in situations like lymphoma. And these affect Fc gamma receptor, both expression as well as function. And so these might affect response to immunotherapies in type 1 diabetes, in the case of drugs like rituximab, anti-thymocyte globulin, and others that have this intact Fc portion in the antibody drug. And we know that in these other disease contexts, that the way that these mutations affect the drug efficacy is through influences on antibody-dependent cell cytotoxicity as well as phagocytosis. So in addition, there's been evidence in other disease contexts that things like anti-drug antibody development can affect drug efficacy. And we know that the human leukocyte antigen region is highly associated with whether someone is going to develop these anti-drug antibodies. So this is something else that would be really interesting to look at in type 1 diabetes, whether there are certain HLAs that might be associated with perhaps the development of antibodies against a drug that might perhaps block their ability to work. And then sort of at the end of the figure, combining all of that together, we can hopefully use artificial intelligence and machine learning to combine all of this information together. So to combine everything that we know about all of these different genetic variants to then inform for an individual person, first of all, what drug should they get, but also what dose of drug should that person receive. So perhaps we might be able to use the same drug on lots of different people, but we may see better response with higher or lower doses. A good example here actually is anti-thymocyte globulin. On the population level, we saw really good responses at a lower dose, whereas at a higher dose, we saw worse outcomes. But when you go in and look at each individual patient at the patient level data, there's a hint that perhaps different doses might be more efficacious for different patients.
Yeah, and again, that kind of follows the tone or the pathway of sort of in the breast cancer world or in cancer in the oncology space where, titration does matter, right? Thank you so much. I love that. I love that way you presented that you sort of the layering approach to this very complex disease. And it really is, you're bringing in some tremendous power from your partnerships with the AI ML piece. I wondered if we might transition a little bit now to Mike and ask about his long-held view of a need for adaptive trial designs and combinatorial therapies.
Sure, thanks.
Wanna
Thanks
weigh
for
in on
letting us speak this morning, Monica.
Sure.
So part of what makes the UFDI so special is the depth and breadth of our scientific discovery approach, as you've seen already from the lovely tour there, and all the work that's being done by folks like Dr. Jacobsen and Dr. Shapiro. And we've long held the view that a single therapeutic is not going to get us to where we need to be in preventing and reversing the disease. And I don't think that's a particularly novel concept, given that most autoimmune diseases are not treated with a single therapeutic, and then you walk away and say you've conquered the disease. But we also have recognized for a long time that the type 1 diabetes is different than treating breast cancer. And the outcomes, unfortunately, for our patients with type 1 continue to get better and better, even outside of the realm of immunotherapies. And so we have to always balance very carefully the potential benefits with the risks of the therapies. And so that's why the work that's being done here is so important, because we really have to figure out which combinations to give to which patients at which time. As the saying goes, the dose makes the medicine or the poison. And much like Melanie was just talking about with ATG, there are going to be patients for whom a low dose of ATG followed by some antigen-specific therapy may be what we need to prevent their disease. And there are going to be others for whom teplizumab and some other immunotherapeutic are going to be more effective. And so we're really just at the very cusp of exploring those things, largely because before the last 10 to 15 years, we didn't have any therapies that really meaningfully augmented disease. And now we have a growing list of almost 10 therapies that can at least preserve beta cell function in type 1s. So that gives some hope to this concept that we can now start to combine those drugs in hopefully rational ways and apply them to very specific populations by understanding their background, genetic risks, and their responses, so that we can move those therapeutics forward in sort of an adaptive trial design. So a concrete example of that is, right now, TrialNet, which we're proud to be, a founding member of, and we've led several of the TrialNet studies, including the one with low-dose thymoglobulin in new onsets. TrialNet is now exploring the idea of having an adaptive trial platform for stage 1 and stage 2 diabetes, with the understanding that we can no longer afford time-wise or money-wise to do a decade-long single study of a single drug. And so what we're seeking to do now is take patients who are stage 1 or stage 2 and put them into an adaptive trial design that will allow us to test at least 3 or 4 different drugs or combinations of drugs every decade, looking for just a signal of benefit. And then if we see it, accepting that we'll have to go on and do a larger study to get a confirmatory result, but knowing that we have to get many more shots on goal in order to move the field forward in a timely fashion. And that's something that, our group is taking a leadership role in helping to push forward. So that's an exciting, place for the field to be, with Dr. Jacobsen now leading the natural history study and really increasing our ability to screen people and put people into the funnel of folks who are eventually going to get type 1. Hopefully we'll have more folks available to do studies, because at the end of the day, as a clinical trialist in the type 1 space, the single thing that has frustrated me in our field the most is that we just don't have enough folks identified who are willing and able to participate in these studies so that we can answer all the questions that have been developed, by our basic science folks and our translational science folks. So,
Can
so
I
yeah,
ask a quick question? You're the site PI for TrialNet. So like, UFLR is acting as the hub for the regional trials and industry-sponsored trials. Can you just talk a little bit about the critical need for collaborative science and biomarker development and monitoring in that whole space? As you're working as a team, you're the nexus, but you have all these other players out at a distance. How are you coordinating all of that? And how are you getting, biomarker development to kind of move forward as a wave rather than individual silos,
Yeah, I mean, that is still a challenge in academic medicine, I think. People working in silos, that has never been the way that the UFDI has functioned. I think that's another thing that really differentiates us. You
Yeah.
can see by the MPOD is an excellent example of the desire to, both create programs and share information and samples with the whole world, so that we raise everybody's knowledge of the disease space. In the clinical trials world, I think we exemplify that by way of we're participants in just about everything. We do, pure academic studies through TrialNet and Teddy. We do industry studies when companies come to us with new therapeutics that they want to test in the phase 1 all the way through phase 3 space. And we do investigator-initiated studies that are, homegrown therapeutics that have been thought of and developed right here in Gainesville. And along the way, in all of those studies, we're often thought of as a place that can help develop those biomarkers to help drive the science forward so that we can develop better, more novel, more targeted studies in the future.
Yeah, it does seem that the actual recruitment of patients to participate is sort of like the toughest piece. Is that correct?
I mean, I'm biased because I'm a clinical trials person, but yeah, I think that's really the biggest challenge to moving the field forward. If every single patient who we ever identified who was stage 2 or newly diagnosed felt the obligation to be in a study, we'd be much further towards a true preventative or curative therapies. It's a small fraction of folks who do that. And again, type 1 diabetes, thank goodness, is not a cancer diagnosis, but in the pediatric cancer world, that's standard of care. Every single patient who gets diagnosed with leukemia is going to get some standard of care therapy and be offered some study to move the field forward. We need to get the type 1 community to be in that same mindset and space. And frankly, we haven't been able to offer them things that were all that exciting in the past, but we're now there. And so hopefully that's going to happen. And over the next few years, we're going to see it kind of evolve to where almost every patient does have that experience and they're offered, here's standard of care, but here's standard of care plus an opportunity to move the field forward for both you, your child, and future children.
Yeah.
And
so the pediatric boots on the ground really have to convey that messaging.
yeah, and Monica, if you don't mind, I could
Yeah.
just like feel like the vibe from Todd that we should also mention that a big trouble with clinical trials is we design these what we think are really rigorous trial designs, but we don't at the beginning set up funding for the mechanistic analyses. So sometimes we're on the back end scrambling to understand if a therapy worked, how did it work, why did it work?
Such a great point.
Yeah, so.
Yeah, so just let me say something about that quickly, because I think it's really critical. Monica, I know you're so ingrained in the mechanistic space that you have an appreciation for the need for AI and machine learning being applied to type 1, and it's something that we care about as well. And the only way you're ever really going to accomplish that well is to have core assays and infrastructure in place where you can collect similar data and make those comparisons so that you can link biomarkers with clinical outcome type data.
True.
So it's something that we're working along with TrialNet, other investigators. We had a great meeting at Breakthrough a couple of weeks ago to discuss trying to get infrastructure in place to really bring this to the forefront and make it a priority in the T1D clinical trial space, because it is, you know, you and I work together quite a bit on the adaptive immune receptor repertoire world. T cell and B cell receptor sequences, that's only going to be really useful in a machine learning type space and having comparable data across the spectrum.
Right. And you've got to have it. You've got to get more data is better in that space. I thank you, for all of that, those really excellent points. I did want to kind of circle back a little bit to Melanie. In the little video, there was a little allusion to HyperGator. I didn't know if you could unpack that using a HyperGator and AI to repurpose drugs and, really yield these novel pathways and precision medicine targeted therapies that you, and digital twins even that you alluded to earlier. And just kind of share with, trainees and others outside of the UFDI, what it really is and why is it exciting?
Sure, yeah. Thanks, Monica. So HyperGator is, I believe at this point, the most powerful supercomputer at any university. So we're really poised to be able to do these very, very memory intensive AI type prediction algorithms. In terms of type 1 diabetes right now, I think the idea of digital twins is at the more conceptual stage. So there are definitely people that are trying to make digital twins in terms of the immune system. And I think that we really need to start partnering with those groups to try to learn what they're doing and see how we can start to use some of their software, some of their algorithms for our own purposes. So right now, I think a lot of the AI that we're doing is more, as sort of everyone else has alluded to, is more in the biomarker identification space. So using AI tools to identify biomarkers, for example, from flow cytometry, genetics, single cell RNA sequencing data, things like that, to identify who will best respond to therapies. But the concepts that you're bringing up are extremely, extremely exciting. And I think that they're sort of the future of type 1 diabetes. But what we need to do is first use these more classical AI tools to identify some of these signatures and then feed those into these newer tools that can create models where we can then test therapies in silico to not have to actually go through the process, perhaps, of having one patient undergo therapy after therapy after therapy. Perhaps we can preemptively predict responsiveness through these in silico type of models.
Yeah, it's really an expeditious pathway. There is a quick question here. I'm going to ask it and then I'm going to pivot over to Todd again. Within the context of basket trials, how and at what time point will response or non-response to interventional therapy be defined? Do you see this evolving the same way as we've seen using immunotherapies in cancer? An example, the use of specific objective response criteria for individual patients and tumors.
Yeah, we're right on the cusp of that. It's a great question. And frankly, we just haven't had the tools to affect meaningful change yet, now that we're developing those and have drugs that seem to affect our endpoint of interest, and namely beta cell preservation, C-peptide. We now can see that there are immunologic changes that occur much earlier than those, and even metabolic changes that precede our endpoint. And so by applying all those earlier in the disease process, we should be able to bucket people and start to do basket trials where people go into one direction or another based on hopefully their baseline data before we even touch them with a therapeutic, but certainly afterwards. And we're pushing them in the right direction by knowing, what their response is and which drug makes the most sense for them. So more to come there, but again, back to my initial conversation, like we can't do any of those things until we have a core sort of philosophy from the field that everybody's going to participate in research. Because to do those studies, we need even more patients than we have available right now.
Yeah, well said. I guess I'm going to, at this point, we have about 10 minutes left, pivot to Todd, and I'm going to ask just a little bit about the vision that you have as the new acting director of the Institute for how single cell and repertoire studies are going to transform how we monitor trial outcomes moving forward.
Yeah, Monica, that's a great question. I think, you know, we have two major areas where I think we have a lot of room for growth and development moving forward. The first is really everything that we've learned and really begun to discover out of NPOD. I think we have the opportunity to really, now that we have the tissue and we have some targets, can we validate that these pathways are active in the pancreas of individuals that are developing the disease? And can we actually begin to do some intelligent trial design rather than taking drugs from different diseases and trying to repurpose them without any sort of idea that that signal is active within the pancreas of a type 1. So that's one of the areas. The other is kind of what you mentioned regarding the adaptive immune receptor repertoire. I think this is really going to increase over the coming years in our ability on an individual level rather than a population level to understand the progression of disease and be able to track longitudinally the burden of the adaptive immune response as it progresses over the different stages and in response to a particular agent. And, you know, I mean, it may be both TCR and BCR together, but it also may be that we have to be able to identify antigen-specific cells and understand the phenotype of those cells. So really getting down to the level of single-cell RNA-seq and integrating that type of data over time. And, you know, one of the other aspects that we really care about, and I think we have a lot of opportunity to develop, is just imaging of the entire pancreas and tracking the function of the entire organ. I think one of the aspects that you'll hear about later from some of our other investigators here at UF is that we think this is a disease of the entire pancreas. And I hate to admit that as an immunologist, but I mean, the signal has repeated over and over again, not only in terms of total volume of the pancreas, but also some of the exocrine biomarkers of disease and generalized inflammation across the entire organ. So I think these are areas therapeutically that have not really been tapped into. And I think we have an opportunity here at UF and through NPOD and other efforts to really begin to tap into some of those moving forward.
Yeah, I'm looking forward to Ed Phelps and others illustrate sort of this inherent fragility of the beta cell that they're sort of looking at very, very carefully. I did want to ask, or open it up to our audience. Are there any final questions about NVIDIA or the use of the HyperGator and Gatortron? Happy to answer that. We didn't really touch on the Gatortron. I don't know if you want to kind of.
Yeah, so I'll talk about that briefly. So one of
Yeah.
the first major efforts of UF in terms of bringing HyperGator's power into the medical system here was to take all of our medical records, scrub them of sort of de-identifying information, and then sort of build natural language processing algorithms so that we could have all of our data from the health system in our HyperGator network. So it's really a powerful approach to being able to bring all of that data on a personalized level into the system.
Got it. Just checking for questions. And then I would also say, in terms of the reach across the Florida system, you've got University of South Florida down there with the Teddy base. You've got the DRI Miami. You've got UFDI. How much, is there a lot of cross-pollination across the Florida diabetes ecosystem?
Yeah, so Jay Schuyler, I can see that he's online. I go down to Miami at least once a year and interact with their group. And obviously we have multiple weekly calls with our trial network as well. So it's a pretty integrated group in that aspect, but you guys want to
I think one of the really fun things that we don't talk about enough is like we do diabetes camp across the state. And so people from Miami and Tampa and everywhere come, and we've done research studies at camp, really like getting those kids involved. And I think they really enjoy being a part of that. And that's where we do a lot of our trial net screening as well. But yeah, I mean, we have collaborators, Sherry down in Miami helps us with statistics, and we're really fortunate with the entire group in the state of Florida.
Yeah. I mean, it's a real call, I would say, to those who are interested in working in the endocrinology, pediatric space focused on type 1 diabetes, whether it be an MD, PhD, or an MD, or just an academic researcher, to really explore the UFDI more closely. And this is a chance we've had a flyover here. And understanding what's going on down there, and all the great things that are happening, all the tools and the collaborative pieces that are already in place. And so I think it's just it's a really incredible opportunity there to work and get cut your scientific teeth in the world of gators, I guess, just sort of stealing that from your video earlier. If there's no further questions, one more question, anyone? Last call for questions. We will post this recording on our website and on our YouTube channel, where we typically have about 1,000 to 2,000 views within the first week. So we're hoping that this will get out and get people very excited about UFDI, as we are. And I thank you again for taking the time out of your busy schedules to really come talk through what's happening there and what the opportunities that exist are at UFDI. Thank you once more to all of you.
Well, thank you, Monica. Yeah, we appreciate all your efforts as well. So thank you.
Thanks. Reciprocal. Have a great rest of your day. We'll look forward to the next two panels. Bye-bye.